I'm going to let you take the spotlight. I'll turn myself off so that you're the only one on screen with your presentation. And
Sure.
then if there's time for questions at the end, I hope you don't mind me popping back on and
No,
doing
not at
some of that.
all. Yeah, feel free
Awesome.
to also interrupt me at any time if you have a question on a particular slide or piece of data. So,
Perfect. So thank you. Thank you. Go ahead. Take it away.
okay, yeah. So, hi everybody. Thanks for joining the talk today. I'll be talking about my current research. Like Dr. Soares said, I'm a fourth-year PhD student in the Biological Sciences department. And I specifically work with brown fat tissue. So today I'm going to be talking about how we investigate different genes that influence mitochondria function and the preservation of healthy fat. So just to start it off a little bit about my lab, my advisor is Dr. Shiwan Kwong in the Animal Sciences department. And our lab is really kind of fun because we work with skeletal muscle and adipose tissue at the same time. And the basis for this is that skeletal muscle stem cells are heavily influenced by adipose tissue, both in their formation and regeneration. So one reason we study the adipose tissue function is to basically look at how that influences skeletal muscle health and development over time from a young age and then as we age into adults. Particularly, I look at the molecular regulators of brown adipose tissue function within the lab. And the thought here is if we can further characterize brown fat tissue and understand genes that regulate it, then we can potentially develop therapeutic targets down the road to preserve healthy fat within our bodies. So with that, I'd like to give just a little bit of background on what brown adipose tissue actually is, because I think for the general public, even myself, before I got into this research, I had only ever really heard about, white fat, that really unhealthy subcutaneous fat that we find. So here we just have a diagram of a mouse showing the three main types of adipocytes. So brown adipocytes, white adipocytes, and beige adipocytes. And I'll start with the main two first. So brown adipocytes have what are called multilocular lipid droplets. And lipid droplets are going to be these small little organelles that store energy in the form of triglycerides. So brown adipocytes have several lipid droplets. But what makes brown adipocytes unique is that they are densely packed with mitochondria. And these mitochondria are actually what give the brown adipocytes that really rich red color that you'll see in future slides. They are highly vascularized and innervated, and they have the unique ability to perform a process called non-shivering thermogenesis, which is quite a phenomenon because it allows us to dissipate energy as heat. So you've probably heard of the term ATP before. This is basically how our body stores energy in the form of fat. And so instead of leading to the production of more ATP for storage, the brown adipocytes actually allow us to burn this energy off as heat, which is very interesting, and I love it. Yeah. So then we have white adipocytes, which in contrast have these unilocular lipid droplets, so singular lipid droplets. They have very few mitochondria, and they are the main type of adipocyte that stores and releases energy, and that energy storage is also in the form of ATP. Now, where things get really interesting is when we start to talk about beige adipocytes. So these can develop in white adipose tissue, and they start to resemble brown adipocytes because they have, again, these multilocular lipid droplets. We see the dense packing of mitochondria, and they are actually recruited and can kind of transform from this white adipocyte identity into the brown adipocyte identity if they're recruited by a cold stimulus or other environmental stressful stimulus. So that kind of gives them this phenotypic plasticity. And so why this is really interesting is because a lot of research can be done to determine how can we kind of promote more white adipocytes to undergo this beiging process and take on this characterization and identity of brown adipocytes, which is the healthier form of fat within our bodies. And the last thing I'll say here that I will explain in future slides is this little protein here called UCP1. It is unique to brown adipose tissue and also these beige adipocytes when they start to undergo that plasticity. UCP1 stands for uncoupling protein 1, and it essentially allows the brown adipocytes to undergo non-shivering thermogenesis and dissipate energy as heat. So I'll be talking about this protein a lot in the future slides, so I just wanted to point that out. So obviously I'm showing you the mouse diagram here, but how does this actually relate to humans? So what's really interesting about brown fat is that we have a large amount of it as newborns. And to put this in context, when we're in our mother's womb, we are used to, a constant temperature, that same environment. But as soon as we're born and we enter that external environment, we're going to be subjected to a lot of temperature changes. So the brown fat is really vital here to help newborns regulate their body temperature and dissipate some energy as heat in order to keep them really warm. And it used to be thought that we only utilized brown fat as newborns, but more recent research has actually shown that over time, as we age into adulthood, we don't have as much brown fat, but we still have a lot of depots of it that can be utilized and are actually really important for us to expend energy instead of storing it in the form of fat. So again, another really crucial reason that people are doing this research in brown fat is to find ways to preserve this brown fat as we age and maybe make more use of it in order to combat obesity and certain metabolic disorders, specifically as we age. And so to give you a little bit more understanding of how this non-shivering thermogenesis works, I want to break down the word first, maybe to help it make more sense. So if you think, let's imagine that we're standing outside in the winter and we're shivering. That shivering effect is coming from our muscles, right? So when we say non-shivering, it's just thermogenesis, the creation of heat that's occurring when we're not, shivering. So this is kind of like a silent little process that's occurring in the brown fat. And so to put things simply, in our brown fat tissue, in response to a cold environment, so again, if we're outside on that cold winter day, there's going to be release of norepinephrine, and it's going to travel to the brown adipose tissue cells, the brown adipocytes, and basically it's going to lead to this signaling cascade that releases free fatty acids from those lipid droplets. So basically, we are breaking down triglycerides in order to try and build back more energy to help keep us warm. Those free fatty acids will travel to mitochondria, and typically they enter the electron transport chain and are used to help generate ATP, so to help generate that energy that we use for storage. But the phenomenon here is that in the presence of this cold stimulus, that UCP1 protein is activated, and it's called uncoupling protein 1 because it uncouples oxidative phosphorylation, which is basically what's shown by this respiratory chain image here. It uncouples oxidative phosphorylation from ATP production. And so instead of generating the ATP, we basically dissipate that as heat. So this is, the main basis of non-shivering thermogenesis. And I just want to reiterate again that this occurs specifically in the mitochondria of brown fat tissue, and essentially relies on activation of this UCP1 protein. So that's a little background on that. Main takeaways here: non-shivering thermogenesis occurs in mitochondria and requires expression of this UCP1 protein. All right, so that's a little background on that. And so again, just to reiterate why brown fat is important for energy homeostasis, a simpler way to put this is, for example, calories in versus calories out. We know that if we consume more calories than we burn, we're going to put on weight. But if we burn more calories than we consume, we're going to lose weight. And so we can also think of this in terms of white adipose tissue and brown adipose tissue. So in the instance of more energy intake than expenditure, we're going to have excess energy in the form of ATP, which will be stored in those white adipocytes. However, if we have more energy expenditure than we have energy intake, we are going to be utilizing those brown adipocytes more and burning off that energy as heat. So this is kind of the delicate balance that we have between these two types of adipocytes. And more broadly, I'm sure we all know that we kind of have a global epidemic in terms of obesity. So every day it becomes even more important to find potential therapeutic targets or avenues for preserving healthy fat in humans and kind of combating obesity overall. Okay, so then I kind of want to talk a little bit about my protein of interest and how we initially became interested in it and why I'm actually working with it. So the protein I'm working with is called LETMD1, and I will show in a later slide what this actually stands for because it's kind of a long name, but sometimes we refer to it as LETMD if that helps you. And so basically how we initially became interested in it is by looking at publicly available RNA sequencing data. So basically this data looks at the mRNA expression of several genes, and you can select your data to look at certain tissues of choice. So here we were looking at brown adipose tissue, which is abbreviated as BAT. And we were looking at two types of white adipose tissue, IWAT and EWAT. IWAT stands for inguinal white adipose tissue. I like to joke with my family and say it's kind of like the love handle area to give them a better idea of what I'm talking about. And the epididymal white adipose tissue, so EWAT, that's kind of like in the pelvic region. That would be where we find that white adipose tissue in humans. So we were looking at these types of adipose tissue, and we wanted to see how the expression of all of these different genes was in these different white adipose tissues. And we have a heat map down here, a little scale bar. Basically what we found is that LETMD1 expression was enriched in brown adipose tissue in comparison to these white adipose tissues. And in addition, we generated some other data that basically showed us a correlation between UCP1 expression and LETMD1 expression. So if you'll remember, UCP1 is activated during non-shivering thermogenesis. So because these expressions correlate, it kind of told us, okay, so that must mean LETMD1 expression is also elevated during non-shivering thermogenesis. And so we wanted to look at this a little bit further. So we ran something that's called a Western blot. And basically this allows you to look at protein expression at a given time in a given tissue. And so we dissected mice and we looked at the protein expression of LETMD1 and UCP1 at both room temperature, which is the RT here, and after cold treatment. So after we subject mice to a cold chamber that's set at about 40 degrees Fahrenheit. And what we saw is that the expression of LETMD1 and UCP1 correlated not only at room temperature in the brown fat, but also after cold treatment. But if you look at the IWAT, we only see UCP1 expression after cold treatment, but we don't really see LETMD1 expression. So basically what this told us is that, once again, the expression of these two proteins is correlated, and it led us to hypothesize that LETMD1 is required for cold-induced non-shivering thermogenesis in brown adipose tissue, which was really interesting. So we kind of took a step back and looked at the literature to see what is known about this protein so far. And just as a reference, here's the full name of LETMD1. It's a mouthful. Leucine zipper, EF hand transmembrane, domain containing protein 1. So we'll just call it LETMD1. But basically what was known about it at this time when we began this research? Basically, it had been identified as a cervical cancer oncogene. So it had mainly been studied in cancer, specifically in breast cancer. And aside from that, there was a little bit of research that had been done in macrophages, specifically relating to immune responses but that was about it. Nothing had really been studied in brown adipose tissue. So knowing that, we wanted to see where is LATMD1 actually located in mitochondria. So within the organelle specifically, there are several locations where a protein could be located. And here's just a little diagram to show you the kind of a cartoon layout of a mitochondria. We have an outer membrane, an inner membrane, and then the space that lies between those two is called the intermembrane space. And then one really, I think it's a beautiful feature of mitochondria is that the inner membrane kind of folds in on itself to create these inner folds called cristae. And so within the cristae, we have one more area called the matrix. And so basically we did an experiment that allowed us to isolate different portions of the mitochondria and kind of destroy certain parts of the membrane with each treatment that we applied to our isolated mitochondria in order to see where LATMD1 is located. And what we found overall is that we believe it's located on the inner membrane of mitochondria. So just to let you know, moving forward, we had this in mind thinking, okay, this protein's on the inner membrane. How could it affect non-shivering thermogenesis from this location? So in order to study this, having all of this information, we wanted to generate what's called a global knockout. So when we say global knockout, we mean a whole body deletion of the LATMD1 gene. So in the liver, in the white fat, in the brown fat, in the kidney, in the heart, every single tissue, we were able to generate a deletion of this gene. And we use something called CRISPR-Cas9 sequencing for this. Long story short, it's a gene editing tool that allows you to target specific introns of a gene and basically delete every exon that falls between those two introns. So for example, from this diagram, we targeted the introns between exon 2 and exon 8. So we deleted the entire portion of we deleted that entire portion of the LATMD1 gene. And as you can see from this diagram down here, this is the normal gene for LATMD1. But when we triggered this deletion of exon 3 through 7, it resulted in a frame shift and a truncated protein that was only 104 amino acids long. So we heavily shortened this gene and we took out a really important portion of it called the transmembrane domain. So basically, this led to the global deletion of LATMD1. So it would no longer be expressed in all of the tissues in our knockout mice. And so moving forward, when you see this nomenclature here, LATMD1 with the superscript knockout, I'm just referring to the mice that have a global deletion of LATMD1. And so as you can see from this panel over here, this is just showing that we validated the knockout. Again, here on the right, we ran a Western blot to look at protein expression. And so when you see this band, it means that protein is present. So as you can see in the wild type samples, we see the protein, but in the knockout samples, we do not. All right, so having done that, we allowed our mice to age until they were 2 months old. This is a typical time at which you study the mice for specific gene deletions and look at their tissues and things like that. So once our mice reached the age of 2 months, we dissected a few initial pairs. And I'll never forget the day, because as soon as we dissected the mice and looked at the brown fat tissue, we kind of knew we had a really obvious phenotype. If you'll remember me saying, the mitochondria make the brown fat really red and rich in that red color. But as you can see in our knockout mice, the brown fat appears white. And especially when you compare the color here with the two white fat tissues shown here, the IWAT and the EWAT, they look pretty similar. So we had a color change. That was huge. And so from this initial dissection, we split up the tissue into different samples to run different experiments. And one of those experiments is something we call paraffin sectioning, that basically allows us to fix a small portion of the tissue, cut it into very thin slices, and apply some stains to it that allows us to look at the histology of the tissue overall. And so here we have the brown fat, the white fat, and the two white fats here. And then we have our wild type mice and our knockout mice in these side panels. So what you can see when you compare the brown fat tissues is the significant contrast in terms of how the tissue histology looks. Specifically in the knockout mice, we have brown fat whitening, and we also have what's called lipid droplet accumulation. And so if you'll remember, the white adipose tissue, as I mentioned, has the unilocular lipid droplets, and that's kind of the phenotype we're seeing here in the knockout histology as well. So that was very significant to us and very telling. And then we also ran another Western blot to look at protein expression of UCP1. And what we could see in our analysis is that the expression of UCP1 is decreased in the knockout mice in comparison to their wild type litter mates, which told us, okay, there's less UCP1 expression happening when we have a global deletion of LETMD1. So maybe there's less non-shivering thermogenesis occurring overall, which was really interesting. And because of this, because we know non-shivering thermogenesis is important for cold exposure, required during cold exposure, we then asked ourselves, are these mice even able to tolerate cold exposure? So in order to study this, we ran a few cold exposure experiments. And when I say cold exposure, let me just set the scene for you. In our lab, we have what's called an environmental chamber. Basically, it's a machine that you can set a timer on, let's say for, 5 days, 10 days, 1 month, whatever. And you can also set your temperature of choice. So the first experiment we ran is called a chronic cold exposure. We call it chronic because it's 1 week long. So we put the mice in the chamber for 7 days. And we set the temperature at 6 degrees Celsius, which is roughly 40 degrees Fahrenheit. So pretty cold, not something I would personally be comfortable living in for a while. But basically, we put the mice in the chamber and we monitor their body temperature over that 1 week period. So on the first day, we take an initial temperature at 0 hours, at half an hour, every hour for the first 6 hours. And then day 1 through 7, we just do 1 temperature reading per day. And what we saw is that there was really no significant difference in the average body temperature for our wild type and knockout mice over the 1 week period. So no change there. We dissected these mice, however, and saw an even more drastic difference in the brown fat color, as you can see here. In the wild type mice, we have that really rich red color. And in the knockout mice, the brown fat is basically completely white at this point. And again, we did paraffin sectioning and staining of the tissue. And again, we see increased lipid droplet accumulation in our knockout brown fat. However, we don't really see a difference in the white adipose tissues. And so one thing I should also mention is obviously during the chronic cold exposure, it's 1 week long. So the mice have full access to food and water. But based on these results, we then decided to run what's called acute cold exposure, which is a much shorter cold exposure experiment. It's only 6 hours long. And during this time, the mice do not have access to food because we want to see how they acclimate to the cold environment without immediate access to food and nutrients. And what you can see here in this bottom panel is, again, another graph. The first 6 hours of that acute cold exposure experiment, we monitor the body temperature of the mice. And what you can see is that by hour number 3, there's a drastic decline in the average body temperature of our global knockout mice. And it falls so rapidly that by hour number 4, the average body temperature fell below 30 degrees Celsius, which is very cold and not safe for the survival of the mice. So we actually stopped the experiment for them at hour number 4 and basically determined that they failed acute cold exposure without food. to set the scene for you a little bit, the mice were very lethargic. They were shivering very furiously. And we actually had to kind of like hold them and warm them up after the experiment was over because they were just that cold. So we could really tell that the cold environment affected them in this instance. And then we kind of wanted to see if we could visualize heat production in the mice after this acute cold exposure. So here I'm just showing a comparison in some infrared thermograph imaging of mice at room temperature and after that 4-hour cold exposure. So basically we use a camera that allows us to look at heat production throughout the entire mouse body and specifically in the brown fat region, which is, I don't know if I have a laser, maybe I do. So the brown fat region is right here, kind of behind the shoulders. And so if let's start with the room temperature first. If we compare the wild type and knockout mouse, you can see increased heat production in the brown fat region for the wild type mouse in comparison to the knockout mouse. There's still a significant difference that we can see by eye by using the scale bar here. But what's even crazier is when we move over to the 4-hour cold exposure, again, we see heat production in our wild type mouse and in the brown fat region. But in the knockout mouse, there's virtually no heat production throughout the entire body, and we see nothing in that brown fat region. So this correlates and kind of supports the result we saw from that acute cold exposure when the average body temperature drastically took a nosedive at hour number 3. So basically, again, the mice cannot tolerate cold exposure without food. And so this kind of led us to start thinking, okay, this LETMD1 protein must be really important for successful non-shivering thermogenesis to occur in a cold environment. And so, yeah, this led us to then ask, sorry, let me get rid of my laser. It's kind of in the way. This led us to ask, we've done a global knockout of LETMD1, but what if we specifically look at it in the mitochondria? Because they're not able to tolerate that acute cold exposure, is anything actually happening in the mitochondria in this global knockout? And so to set the scene for this, I kind of want to introduce the technology we use called transmission electron microscopy. It's amazing. It's one of the favorite things I've learned in grad school how to do and be trained on. But basically, we use this instrument that shoots a very high powered beam of electrons through a very thin section of your tissue of choice. And because we're shooting a beam of electrons through the tissue, it then allows you to view through your microscope your actual tissue and your organelle of choice, if that's what you're choosing to look at. So just to show you how this compares with a 3D image of a little cartoon image, of mitochondria. Again, we have the outer and inner membrane with that intermembrane space in between. We have the Christie structure, and then we have the matrix that's inside of those Christie folds. And how this correlates with the TEM image, you can see here on the right, we have that nice outer membrane. The inner membrane is a little bit harder to see, but you can clearly see these Christie structures. They kind of look like noodles, if that helps you think of it, that kind of just run across the entire mitochondria. And so when we see this on a TEM image through our microscope, we know that we're looking at mitochondria. So we used our wild type and global knockout brown fat to look at the mitochondria structure through TEM imaging. And just to show you here on the left, here's our wild type sample. When we zoom in on the image, you can see we have these really nicely formed mitochondria with these beautiful Christie structures that span the entire width of the mitochondria. They look healthy, they look good. And if you kind of squint a little bit up here, you can see that the cells are so densely packed with mitochondria, which we expect because we are looking at brown fat tissue. However, when you look over here in the right panel at the knockout mice, immediately you can see how fewer mitochondria there are throughout this cell. And then when we zoom in on a little section, what we notice is that these mitochondria appear completely empty they appear darker in color, and we really don't see Christie structures at all. They appear to just be gone. And so we actually were able to quantify this. Basically, we took several images, and in each image field, we counted the number of mitochondria, but also the number of cristae per mitochondria. And what we found is that for the knockout mice, both of those values were significantly decreased. And so because of this, again, we're still in our global knockout, but we wanted to look more specifically at adipose tissue to be sure that deletion of LETMD1 was kind of specific to the brown adipose tissue, and that's why all of this phenotype was occurring. So in order to look more specifically at tissue-specific defects in adipose tissue, we generated something called a conditional knockout. And so if you'll remember, I said a global knockout is in every single tissue throughout the entire mouse and throughout the body. What's really cool about a conditional knockout is you're basically setting a condition. So this type of genetic engineering allows you to delete a gene of choice in a specifically targeted organ, tissue, or cell. So in our case, we decided to delete LETMD1 in adipose tissue. So in white adipose tissue and brown adipose tissue. And here I'm kind of just showing the scheme for that deletion. This requires a little bit more, how should I say? It's a little bit more difficult to generate these mice, not only in terms of breeding time, but in terms of the generation of the deletion. I won't explain too much because it can get a little bit confusing, but we use something called Cre-Lox recombination. And basically this system allows you to place something called Cre recombinase at the promoter region of a gene of choice. And typically you choose a gene that's only found in your tissue, cell, or organ of choice. And so we chose AdipoQ, which is found in adipose tissue. And basically, when this Cre is turned on, we also have two other components called LOXP that surround LETMD1 and our exons of choice. And so when Cre is present, and we also have those two LOXP sites present, it leads to the deletion of your specific gene region of choice, resulting in your conditional knockout. So again, this AdipoQ Cre knockout that we generated is a specific deletion of LETMD1 in all adipocytes. And so for the rest of the PowerPoint, instead of showing the superscript knockout, you will probably see that I have the AKO superscript, which refers to the AdipoQ Cre conditional knockout mice. But again, the phenotype really did not change. Not much was different after we generated this knockout. As you can see here, upon dissection of 2-month-old pairs, the brown adipose tissue still appears very white and heavily contrasts that dark red color of our control brown adipose tissue. When we ran a Western blot to look at protein expression, you can see here that UCP1 protein expression was still significantly decreased in our knockout mice. And again, when we looked at tissue histology, we still see increased lipid droplet accumulation in comparison to the control brown adipose tissue. So knowing that, again, we wanted to go back to the TEM imaging and see, is the same thing still happening as far as mitochondria structure and cristae formation? And what we saw is actually a more severe effect on the mitochondria structure in this conditional knockout. So here on the left, looking at our control mice, so mice without this conditional knockout of LETMD1 in the adipose tissues, we still see those really beautiful, nicely formed mitochondria with cristae that span the entire width of the mitochondria. However, when we look at the conditional knockout mice, we actually see mitochondria morphologies that still contain the cristae structure, but are a little bit more severe in how they look. So there have been several studies that talk about this mitochondria morphology. And something I'm going to talk about in the upcoming slides is mitophagy. which is basically the detection and destruction of unhealthy or damaged mitochondria. And what studies have found is that in the process of mitophagy, mitochondria actually take on these different morphologies or shapes when they are damaged or unhealthy before they are destroyed within the cell. And two of these morphologies are called swollen and donut shaped. So let me just back it up here to show you. This swollen morphology, when this takes place, the cristae kind of fold in on themselves and create these little pockets or circles in the inner part of the mitochondria. So that's shown here with the pink tags. And then the donut-shaped mitochondria, exactly how they sound, we either have a single circle of cristae within the inner part of the mitochondria, or they kind of appear empty and void of the cristae altogether. But here I'm just pointing out two of those morphologies. And this really interested us because we didn't see these in the global knockout images, but we're seeing them in the conditional knockout images. So we quantified these as well. So again, we just counted the number of mitochondria, and specifically we looked at the swollen morphology, the donut-shaped morphology, and the normal morphology. And what you can see is that those morphologies indicative of mitophagy were significantly increased in our conditional knockout brown fat, whereas the normal healthy looking mitochondria were significantly decreased in those conditional brown fat. So this was really interesting and led us to start thinking about mitophagy and what we could actually be capturing in these conditional knockout mice. So I just want to give a little background on mitophagy and explain what I'm actually talking about with that. So this is a small little diagram explaining mitophagy. So again, this is the selection and destruction of damaged mitochondria within a cell. You may have heard the term autophagy before. So phagy of self, kind of death of self. Mitophagy is a form of autophagy. We just stick the mito tag on the front of it to directly refer to mitochondria. So what's happening here? We have damaged or unhealthy mitochondria that are not functioning properly, not structured properly. It could be a variety of things. When this happens, these damaged mitochondria are recognized by the cell through different receptors and other factors. And when this happens, there is something called an isolation membrane that begins to form around the mitochondria, basically targeting it and selecting it because the cell knows, hey, this mitochondria is damaged, we need to get rid of it. So this membrane will start to form around the mitochondria, and eventually when it's fully formed, we call this an autophagosome, so self-phagosome, and that fully forms around the mitochondria. Eventually what happens is that this autophagosome is going to fuse with a lysosome. And so within cells, lysosomes are these really interesting organelles that basically have a lot of enzymes within them that can degrade and get rid of any damaged organelle or other component that's floating around within a cell that's that needs to be destroyed. So the autophagosome will fuse with the lysosome, and then eventually that damaged mitochondria will be degraded within the lysosome and gotten rid of. So this is basically what mitophagy is. And we believe we're seeing signs of this in our conditional knockout mitochondria. That's kind of the take-home message here. It's really interesting to me because we can actually visualize this with TEM imaging. So down here, I'm showing a control sample. So basically mice without a conditional knockout of LATMD1. And again, you can see that these mitochondria appear undamaged. The cristae structure looks beautiful. Everything's going well. But then what we have capture in another conditional knockout that we're working with is in real time, kind of these different steps of mitophagy. So here in the second panel, I want to show the autophagosome formation. So again, we have that membrane that forms around the mitochondria. And here the red arrows are indicating, if you look closely, you can kind of see those mitochondria and the fact that they're being surrounded by this autophagosome. Then we've also been able to capture fusion of that autophagosome with the lysosome. So you can see this really nice kind of pinched area here in this image. And we believe that that is the autophagosome containing damaged mitochondria fusing with a lysosome. And then finally, we have just full-blown degradation of mitochondria within the lysosome. So once this is happening, we don't see that nice circular structure of the mitochondria anymore. It's kind of just, like I said, being degraded. There are a lot of enzymes in the lysosome that help take care of that. And we believe we've been able to capture that with one of our conditional knockout models as well. So that is really interesting to us. And that's kind of where we're at right now. Obviously, we've established a phenotype for LATMD1, but it's kind of the easier part of research, I would say, because now we're in the uphill battle of determining how LATMD1 actually functions. And with that, we're still trying to determine if mitophagy is just a secondary effect of LATMD1 deletion, or if LATMD1 function is directly related to mitophagy. So it's a really interesting kind of dilemma, but puzzle that we're trying to solve right now. And so just to give a little schematic for what I've talked about today, here on the left, we have a wild type mitochondria, and kind of just showing that when there is a cold stimulus or when the mouse is in a cold environment, this leads to that norepinephrine release on the cell membrane of mitochondria in brown fat. It leads to the UCP1 protein expression and UCP1 activity, and non-shivering thermogenesis occurs as normal. However, in our global and conditional knockout mice, what we're seeing happen is that when there is a cold stimulus, UCP1 activity is actually significantly decreased, and non-shivering thermogenesis is not able to occur. And in conjunction with that, we have loss of normal cristae structure and mitochondria function overall. Some of the data I didn't show today, but I can just explain a little bit. Not only are there less mitochondria and cristae per TEM image field, but we just have less mitochondria overall, both at room temperature and after cold exposure. So we're also working with the fact that these knockout mice don't have as many mitochondria. And so that's kind of interesting to think again about how LATMD1 functions in that situation. So next steps overall, after we have determined and believe that LATMD1 is required for BAT thermogenesis and mitochondria structure, we need to characterize it and how it actually functions on the inner membrane. Like I mentioned at the beginning of the PowerPoint, the better understanding we have of brown fat, the better we are able to kind of manipulate it and potentially create therapeutic targets for people that are facing metabolic disorders. And the process of characterizing this gene will be identifying protein candidates that potentially interact with the LATMD1 protein on the inner mitochondrial membrane. Because once we determine potential proteins that it interacts with, this could tell us more about how it functions and potentially lead to a mechanism overall for how we believe it influences brown fat thermogenesis and mitochondria structure. So that's kind of the part of the research that I'm in now. And so that, obviously, I've got to have my acknowledgement slide. First of all, my advisor, Dr. Kwong, is great. I'm so grateful that he's given me the opportunity to work with brown fat in this project. Two former postdocs in our lab, Dr. Feng Ye and Dr.